Lipids are generally known as oils and fats and are substances which are insoluble in water. All the living cells have two phases, the aqueous phase and the lipid phase in their cytoplasm. The biochemical and biophysical reactions in a cell occur in one of these two phases. The lipid content of tissues varies widely. Rapidly growing embryonic tissues have about 1 to 2% of total lipid, while tissues in which lipids are stored may contain 90% of lipids. Lipids are extracted from the tissues and cells by non-polar solvents like chloroform, ether or benzene. Lipids can occur either as free molecules or in bound form as glycolipids combined with carbohydrates or as lipoproteins combined with proteins. The lipids play a very important biological role in our body. Their major functions are as energy source. Lipids act as fuel in the body and yield 9.5 kilocalories per gram energy upon oxidation. As storage compounds, since lipids are insoluble in aqueous solution, they can be stored easily in body almost in unlimited amounts. Insulation: fats are usually stored in the subcutaneous layers and exert insulating property. Lipids are also stored around internal organs and are found to be padding and protecting the organs. Structural role: phospholipids and cholesterol are important components of lipid bilayer in membranes. Endocrine function: hormones like adrenocorticoids, mineralocorticoids, sex hormones are synthesized from lipids like cholesterol. Vitamin absorption: lipids are essential for the absorption of fat soluble vitamins like vitamin A, vitamin D, vitamin E and vitamin K. Nervous system: lipids play a vital role in nerve cell insulation and impulse conduction. Antibiotic activity: squalene, a steroid from shark's blood, has been found to possess antifungal and antibacterial activity. As cell surface components, lipids participate in cell recognition, species specificity and tissue immunity. Now we come to classification of lipids. Lipids are classified on the basis of their backbone structure as complex lipids and simple lipids. Complex lipids contain fatty acids and simple lipids do not contain fatty acids. We need to know about fatty acids before understanding lipids. Fatty acids have a long hydrocarbon chain attached to a terminal carboxyl group. The hydrocarbon chain may or may not have a double bond, and fatty acids are classified accordingly as saturated with no double bond or unsaturated with one or more double bonds. A simple formula is used to indicate the length of the hydrocarbon chain and the position of the double bond in a fatty acid. A saturated fatty acid like palmitic acid with a 16 carbon long chain and no double bond is written as 16:0. An unsaturated fatty acid like oleic acid with an 18 carbon chain and one double bond is written as 18:1 delta 9, indicating the position of the double bond at the 9th carbon atom. An unsaturated fatty acid like linoleic acid with same number of carbons but two double bonds is written as 18:2 delta 9 12. Fatty acids with 16 and 18 carbon atoms are the most common. Some of the fatty acids are synthesized in the body, while a few others needed for normal metabolism are to be taken in the diet. The fatty acids needed in the diet are referred to as essential fatty acids. They support brain development and functioning, cardiovascular health, skin health, etc. oleic acid, linoleic acid, linolenic acid and arachidonic acid are some of the essential fatty acids. Sources of these fatty acids are vegetable oils like soybean, sunflower, safflower and corn oil, nuts like almond, avocado and walnut, green vegetables like spinach and broccoli and animal products like meat, eggs and fish. The complex lipids have two types of backbone structure made up of either glycerol or sphingosine. Fatty acids are attached to one of these compounds. The first category amongst these is triacyl glycerol. Glycerol has 3 hydroxyl groups, and when all the 3 groups are esterified with fatty acids, they are called triacyl glycerol or triglycerides. The triglycerides are called simple triglycerides when all the 3 hydroxyl groups are occupied by the same fatty acid. For example, in tripalmitin, all the 3 hydroxyl groups are esterified by palmitic acid. In triolene, all the 3 groups are esterified by oleic acid. The triglyceride is a mixed triglyceride if the 3 hydroxyl groups are esterified by more than one type of fatty acid. For example, 1-palmitoyl diastearoyl glycerol has 1 molecule of palmitic acid and 2 molecules of stearic acid esterifying the 3 hydroxyl groups of glycerol. Triacyl glycerols are the most abundant family of lipids and are the major components of storage lipids in plants and animals. Triacyl glycerols, which are solid at room temperature, are called fats, and those which are liquid at room temperature are called oils. Fats have different characteristics, and plant and animal fats differ from each other in some physico-chemical characteristics. Animal fats are rich in saturated fatty acids, while plant fats in unsaturated fatty acids. Animal fats are solids at room temperature, while plant fats are liquid. In animals, fats are stored in liver and bone marrow. In plants, they are stored in seeds and fruits. Examples of animal fat includes butter fat and beef fat, and the plant fats are olive oil and castor oil. Diacyl glycerols with only 2 of the 3 hydroxyl groups esterified by fatty acids and monoacyl glycerol with only 1 hydroxyl group esterified with a fatty acid are also found in smaller quantities in nature. The next category is ether acyl glycerol. Besides triacyl glycerol, there is another family of glycerol lipids, the alkyl ether acyl glycerol, which are less abundant in occurrence. Here, the 2 hydroxyl groups of glycerol are esterified with fatty acids, and the third hydroxyl group is joined in ether linkage with a long alkyl or alkenyl chain. The third category is glycosyl acyl glycerol. This group contains a sugar in glycosidic linkage with the unesterified 3 hydroxyl group of diacyl glycerol. A common example is galactosyl diacyl glycerol found in higher plants and also in neural tissues of vertebrates. Now we come to the second type of complex lipids known as phosphoglycerides. These are commonly called phospholipids. The phosphoglycerides form the major component of cell membrane. In these molecules, one hydroxyl group of glycerol molecule is esterified to phosphoric acid and the other 2 are esterified to fatty acids, of which one is saturated and another is unsaturated fatty acid. The parent compound is phosphatidic acid, to which a nitrogenous base is attached. The most abundant phosphoglycerides in higher plants and animals are phosphatidylcholine, popularly known as lecithin, and phosphatidyl ethanolamine or cephalin. Besides, there are phosphatidylserine, phosphatidylinositol and phosphatidylglycerol. One type in this group is plasmalogens, in which there is an ether linkage at the first position. These are abundant in membranes of nerve and muscle cells. The phospholipids play an important role in structure formation, enzyme action, blood coagulation, lipid absorption and transport, electron transport and ion transport. The sphingolipids. Sphingolipids are the second type of complex lipids having sphingosine as the backbone structure. They are important membrane components in both plant and animal cells, especially in brain and nerve tissue. A sphingolipid contains a fatty acid, one molecule of sphingosine and a polar head group as chief components. The sphingosine contains a long saturated or monounsaturated fatty acid of 18 to 26 carbon atoms connected to its amino group by amide linkage. The resulting compound has two non-polar tails and is called ceramide, which is the parent structure of all sphingolipids. The sphingomyelins. In tissues of higher animals, sphingomyelins are present, which contain phosphorylethanol amine or phosphorylcholine as their polar head groups. These bases are esterified to the hydroxyl group of ceramide. The neutral glycosphingolipid. These lipids contain one or more neutral sugar residues as their polar head group. Hence, they have no electric charge. The simplest in this class is cerebroside, which contains a monosaccharide bound to the hydroxyl group of ceramide in beta glycoside linkage. The cerebrosides of brain and nervous system contain D-galactose and are called galactocerebrosides. The sulfate esters of galactocerebrosides, known as sulfatides, are also present in brain tissue. Another type is acidic glycosphingolipid, which are commonly known as gangliosides. This group is the most complex of glycosphingolipids. They contain one or more residues of sialic acid, which gives a net negative charge to the polar head of gangliosides at neutral pH. The sialic acid found in human gangliosides, N-acetyl-neuraminic acid. The gangliosides are abundantly present in grey matter of brain and in small amounts in non-neural tissues. Now, let us see the functions of glycosphingolipids. These are involved in transmission of nerve impulse across synapses. They are present at receptor site for acetylcholine and other neuron transmitters. They play an important role in blood group, organ and tissue specificity. They are involved in tissue immunity and in cell-cell recognition. Many genetic deficiency diseases occur due to abnormal accumulation of different glycosphingolipids. Example, in Tay-Sachs disease, gangliosides accumulate in the brain due to genetic lack of enzyme required for its degradation. Another type is waxes. Waxes are water insoluble solid esters of higher fatty acids with long chain fatty alcohols or sterols. These are found on skin, fur and feathers in birds, leaves and fruits of higher plants and exoskeleton of many insects where they serve as protective coating. The main component of beeswax are palmitic acid esters of long chain fatty alcohols with 26 to 34 carbon atoms. Lanolin or wool fat is a mixture of fatty acid esters of sterols, lanosterol and agnosterol. Now, let us see the physiological importance of waxes. The most important physiological function of waxes is as a protective agent on the surfaces of animals and plants. Waxes are found on the surface of feathers and hair, due to which they remain soft and pliable. The waxes prevent aquatic animals from becoming wet. Due to this, water related birds like duck are able to maintain dryness. The waxy coating on the surface of plants protects them from excessive loss of moisture. Hence, desert plants like palm and cactus can live for long periods without rain. Waxy coating helps the plants prevent the infection from fungi and bacteria which can cause diseases. The waxy coating on several fruits like apples and citrus fruits prevent them from drying out, and thus these fruits can be stored for long periods of time. The second broad category is simple lipids or non-saponifiable lipids. The simple lipids do not contain fatty acids. There are three major classes of simple lipids: the terpenes, steroids and prostaglandins. Let us discuss about terpenes. These are made up of multiples of the 5-carbon hydrocarbon isoprene. The simplest terpene contains 2 isoprene units and are called monoterpenes. Sesquiterpenes have 3 isoprenoid units, and di, tri and tetraterpenes have 4, 6 and 8 isoprenoid units respectively. Many terpenes have been identified in plants, most of which have characteristic odor or flavor and are major components of essential oils derived from such plants. Example: the monoterpenes geraniol, limonene from lemon, menthol from mint and camphor. Phytol chain, a component of photosynthetic pigment chlorophyll, is a linear diterpenoid alcohol. The carotenoids, accessory pigments in photosynthesis, are tetraterpenes. They are also precursors for the synthesis of vitamin A through beta carotene. Squalene, an important precursor in cholesterol biosynthesis, is a triterpene. Natural rubber is a polyterpene containing long hydrocarbon chains having hundreds of isoprene units. The fat soluble vitamins, vitamin A, E and K, are all derived from terpenes. Another class of terpenoid compound functions as coenzymes. Example: ubiquinone or coenzyme Q, which functions as hydrogen carrier for biological oxidation reduction in mitochondria and chloroplast. The second class is steroids. Steroids are another type of simple lipids which do not contain fatty acids. They are derivatives of the saturated tetracyclic hydrocarbon perhydrocyclopentanophenothrin. All steroids originate from the linear triterpene squalene, which readily undergoes cyclization. The first steroid product of this cyclization is lanosterol, which is the precursor of cholesterol in animal tissues. Cholesterol is the most abundant steroid in animal tissues. It occurs in plasma membrane of many animal cells and in lipoproteins of blood plasma. Cholesterol is the precursor of bile acids which help in emulsification and absorption of lipid in intestine. The androgens, which is the male sex hormone, the estrogens, which is female sex hormone, progesterone and adrenocortical hormones are also steroids. Higher plants do not contain cholesterol, but contain other type of sterols known as phytosterol, mainly stigmasterol and cytosterol. Fungi and yeast contain mycosterol and ergosterol. Bacteria do not contain sterols. The most important steroids are the group of compounds having vitamin D activity, which helps in the absorption and metabolism of calcium. Now let us talk about prostaglandins. These are family of fatty acid derivatives which have a variety of hormonal or regulatory biological activities. These are lipid soluble acidic substances found in seminal plasma, the prostate gland and seminal vesicles. The prostaglandins lower blood pressure and stimulate the contraction of smooth muscles. It also functions as regulator of metabolism in different ways. All the natural prostaglandins are biologically derived by cyclization of 20 carbon unsaturated fatty acid like arachidonic acid. The prostaglandin are named according to their ring substituent and the number of additional side chain double bonds. Example: PGE1, PGF1 alpha and PGF2 alpha. These in turn are the parent compounds of other biologically active prostaglandins. Now let us discuss the functions of prostaglandins. The prostaglandins differ from each other in their biological activity. All of them show activity in lowering blood pressure and inducing smooth muscle contraction. PGE1 antagonizes the action of certain hormones. PGE2 and PGF2 alpha are clinically useful in inducing labor and bringing about therapeutic abortion. The next category is lipoproteins. Certain lipids associate with certain proteins to form lipoproteins. These are of two major types: transport lipoproteins and membrane systems. In these, the lipid and proteins are not joined covalently, but are held together by hydrophobic interactions between non-polar portion of lipid and protein component. In humans, plasma lipoproteins occur in four major classes which differ in density as well as particle size. The lipids and proteins here occur in fixed ratio. Depending upon this ratio, there are four classes of plasma lipoproteins, namely chylomicrons, very low density lipoprotein VLDL, low density lipoprotein LDL and high density lipoprotein HDL. All four lipoprotein classes have density less than 1.21 gram per ml. The plasma lipoproteins carry water insoluble lipids between various organs through the blood. Once chylomicrons are released into circulation, they travel to the liver where some of the lipids are extracted for use in the liver. Next, they travel to adipose tissue where more lipids are extracted and stored. Then they circulate to various parts of the body where cells take the lipid and cholesterol they need to survive or divide. Finally, they return to the liver so the body can dispose of the excess cholesterol it does not need. The second type of lipoprotein are membrane lipoproteins. Membranes contain 50 to 60% proteins and 40 to 50% lipid. The lipids are present in fixed molar ratio which is genetically determined. Different models of membrane structure have been proposed and the most accepted one is fluid mosaic model, which consists of phosphoglyceride bilayer into which globular proteins penetrate partially or completely. This model postulates that the phospholipids of membrane are arranged in a bilayer to form a fluid, liquid, crystalline matrix or core. In this bilayer, the lipid molecules can move laterally and the proteins are embedded in the membrane either partially, that is extrinsic or peripheral, or completely, that is intrinsic or integral. This accounts for the fluidity, electrical properties and permeability of membranes. Now, to conclude the module, it may be concluded that lipids are classified into complex and simple lipids. They have significant functions in our body. The essential fatty acids and lipoproteins are particularly important for maintenance of biological activities.